To evaluate how closely the organoids recapitulated the human embryonic cortical development, the authors performed immunohistochemistry at different time points in the development of these organoids. So here they are showing day 28. Here is day 56, and this is day 84. So as the time goes by, we can see how the organization and how the heterogeneity of the cells composing these organoids increase. First, we see a lot of stem cells here, and only a few committed neurons, which resembles this stage in here, in which we have multipotent cells and we have a few committed neurons. Then by day 56, we can observe that some of the stem cells started to migrate to the outermost layer of the organoids, and we can observe these nice layers of committed neurons, which looks pretty much like this stage in the cortical development. Again, we have multipotent stem cells here, and we have several lineage of committed neurons here, which stand for these two neural markers. So by the day 84, we have a high complexity in the tissue. Again, we have stem cells in the innermost part of the organoid. Very interesting, these organoids show this separation of cells, which is pretty natural in the cortical development. So we can see it in here. This. We have also committed neurons that resemble this outermost layer in the cortex or cerebrum. So by day 84, there are other types of cells besides neuron. We have these specialized radial glia cells, which are found in the outer ventricular zone. So these organoids were able to self-organize in this outer ventricular zone, which is this one here. So again, stem cells here in the innermost layers. We have this clear separation of cells or layers. Here we have these OSPC cells zone, which is the outer ventricular zone, which is rich in outer radial glial cells, these ones. So these markers, these two, are for those glial cells. I'm gonna write it in here. Glial cells. So we can find them here. So this confirms that the organization of these organoids is really high and can resemble some stages of the cortical development. We have in the outermost layer, we have committed neurons. So yes, they were able to replicate this process in vitro with their process. So after that, we have different cell types. So as I mentioned before, the cortical or cortex tissue, besides being highly organized, is composed of several different cells. We can see here all the different cells that is composed of. So these markers are for these different types of cells or layers. Typically they are 6 different layers here in the normal cortical development, and each layer has a character or has been assigned to a characteristic cell or cell line. So here on day 19, we can observe that the outermost layer is rich in these caudal reticulus neurons, which are shown in here, which are these ones. So we can see how they start to migrate from the center of the organoid to the outermost part. We can see on day 56, committed neurons which express CTIP2, TBR1. This TBR1 is a marker of these types of cells, which we can find in here, which we can find in here, in this part. So, and here also. So these are this SITB2 is another marker for another different type of cells, which is pretty much showing the stratification of the tissue in vitro and how it resembles the superficial layer neurons. By day 70 and 84, these superficial layer neurons are even more prominent. So here we can see that these cells are very well stratified again. So in short, the authors are just trying to tell us that the tissues are very well organized. They were self-organized with the cues introduced by the drugs in the differentiation media. We can see how they start to resemble the cortical tissue in the fetus, and they also show how the thickness of these different layers were changing with the time. Now, to further compare the forebrain organoids to in vivo human brain development, the authors perform RNA-seq. So they compare the organoids from day 26 to day 100 to different tissues in vivo. So here we can see that these organoids very closely resemble the brain and spinal cord in vivo, especially for day 100, which means that these more mature organoids are more closely related to the structure and transcriptome of the in vivo tissue. They also compare the molecular signatures of the organoids to those of in vivo tissue of a specific region of the cortex. Here we can observe that the organoids from day 14 and day 54 highly resemble the in vivo tissue of 8 and 12 weeks post-conception, and the organoids of day 100 resemble more closely later tissue from week 17 to 24 after conception. So here it's pretty much the same case for these three. So we can conclude that the organoids resemble better prefrontal cortex tissue than other region of the cortex, even when it resembles in some way the molecular signatures. So the gene ontology analysis reveal enrichment of many neuronal function pathways among upregulated genes. So we can see here how closely the organoids from day 100 resemble the upregulated genes in the fetal tissue, brain tissue. When they did the analysis to evaluate which genes were upregulated, they observed these neuronal function pathways such as synaptic transmission, transmission of nerve impulsive, etc. From the immunohistochemical assays of the early organoids, we observe that there are some markers of cell communication, such as beta-catenin. So this means that the cells, that the neurons, somewhat can communicate between each other, but the authors wanted to make sure that the electrical activity was normal or close to normal in these organoids. So the authors perform this electrophysiological assay to assess the physiological properties of the cells in organoids. So when the cells, the neurons communicate between each other, they have these action potentials that fire between each other. So when they are well connected, we can see a train of action potentials. So they observe this in the organoids, which means that the cells are actually communicating and that their physiological properties are very close to normal.